Hello everyone, my name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. Today I'm going to start a new series, or I would say a complete advanced level course. In this course we will learn from the scratch to advanced level. We will learn about computer-aided drug designing. Computer-aided drug designing is not as simple to pick compounds and dog and you say that I have learned it. No. In reality, this is a complete field with too many things, and after making some videos I felt, and from your questions I felt that I need to share each and everything with all of you guys in a systematic manner, and therefore I decided to take time and start a kind of course. What will be my aims during this course? You should know about this. I will not only present during my videos, in fact I will dive in a proper teaching mood of explanation. So don't be so much happy, because what eventually it will result, as a result these video series will be longer, but I guarantee you one thing, that if you watch all these videos you will not regret for your time. This is my guarantee to you. So now the ball is in your hand to start this course by watching these longer videos with proper explanation, or just to skip and watch some smaller videos somewhere else. Upon completion, I assure you to be a skilled person in the field of CAD, computer-aided drug designing, because I want you not only to watch my videos, but I also want you to learn something real skill, real science. In the field, you will be surprised that in the field currently almost 80 to 90% are doing only molecular docking and screening, nothing else. This is only, if I would say in percentage, this is only 5% of the computer-aided drug designing. You are skipping 95%, and that is why you remain for most of the time confused. If you finish this course, I also guarantee you that you will be able to run your own big project with real success. So let's start. If you want to be with me, so let's start our part 1. So what we are going to do, first of all I will tell you about some database. This is called ChEMBL database. You can write this name in Google and you can have this database in front of you. Why? This database is very much rich database about the compounds. You can read here 2.4 million compounds it contains. It contains 1.6 million assays. Do you know what are assays? Assays are actually biological experiments, biochemical experiments, sorry, not biological, the term is not very good. So assays are actually biochemical experiments performed in different labs in different parts of the world. So why not take advantage of this assay and make your project more powerful? But right now you don't have anything, you are only listening to me. So how can you generate your project? I assume right now you have a protein in your mind let's say your protein is EGFR, epidermal growth factor. Okay, so what I have opened, this is a database, we called it UniProt. If you don't know, this is a protein database, and you can find information of all the protein, almost all the proteins in this database. So uniprot.org, uniprot.org. Just I will write EGFR and it will give me some result. Okay, this is the EGFR and I will choose the human one. You see EGFR underscore human. So right now, I suppose in your mind you have some protein. Let's say your protein is EGFR, this one. Now what we will do next, come to the ChEMBL and write down EGFR, or also you can write down this number P00533. P00533. You can also write the protein name EGFR, that doesn't matter. Now if you see target here, it means that the ChEMBL contains some assays which uses this protein as a target. Click here, the targets. Now you can see here different results, but to understand these results, some will say you read this section type. This is for single protein, this is for protein-protein interaction, this is for chimeric protein, this is for protein-protein interaction, and so on, protein families. So I think we all will be interested in this one. It says protein-protein interaction here, but here you can also see two different codes, accession numbers of UniProt, but we are only interested in this one, single protein. For this protein, the ChEMBL database contains 14,459 molecules or compounds. It's a huge data, and almost 32,897 activities. It means different labs have targeted this EGFR in different assays for binding, for inhibition, whatever, they use different drugs or different compounds. So first of all, we will learn how to use this data to design our own drug. But as I told you, drug designing is not simply a docking. So don't jump to the docking part quickly. So in this course, we will learn step by step many things. Now you have searched your protein in UniProt, then transport it or retrieve the records of your protein in ChEMBL, and you found some data. If the ChEMBL do not have the data for your protein, it means this course is not for you, although you can watch it, because you can join us in some other step. You can take your data from some other sources, from other databases, or from your colleagues, whatever, and you can use you can come with us in part 2, part 3, etc. But this part is specifically for the ChEMBL and UniProt. Okay, so what is next? Now come to my Colab repositories, Colab Research, Google Colab, GitHub /h100. I will drop all these links in the description section of this video, don't worry. When it is open, you will see a BioPython. You need to click here. and I have created a separate repository CAD project. Click the CAD project. Now you have different options here. So drug designing project part 1, you can see here. So you need to click here. If you want to be so quick, so this repository will be enriched to up to 15 lectures, and you can go before than me, but I would not suggest to go before than me. The reason is you may not enjoy proper learning. Okay, so this is the repository. It tells you about my name and about this channel which you are watching, about the paper from where I got all these materials. You can open it, and also about those people from where I got much more of help, or from where I got most of the materials. Okay, what will be the learning and training objectives? In this section, in this part first, we will learn how to analyze, how to import, and how to understand the data from ChEMBL. Okay, and here I have connected my notebook. You can do the same. Before to start, in every lecture, I will took 5 to 10 minutes. Today I will took just 2 minutes to explain to you some major basic concepts. So for this video, the concepts are the units. You may heard IC50 or PIC50. What are these units actually? So IC50 means inhibitory concentration for at the half-life, or I would say half maximal inhibitory concentration. To understand you simply, I can give you a good example. For example, you have a drug and I am the patient, you are the doctor. So if you have, and the drug says that you need to give me this drug, let's say 3 bottles. If I drink 3 bottles, then this drug will work. In other words, in scientific words, the dosage is 3 bottles. I would say 3 liters to make it more scientific. Okay, if the dosage for that is 3 liters, then it will work. But similarly, you also have another drug, and it will also work, but for that, as a doctor, you will need to prescribe me 1 pill, I mean 1 tablet. So which one you will prefer for your patient? For me, of course, the drug which works in 1 tablet is good. Why? Because it works the same at low concentration. The first drug, I will need to use it for 3 liters, and the second one I will need to use it just say in 1 glass of water, or just 1 sip of water, and it will work. So of course it's a common sense. The same way, if the IC50 value is lower for a drug in any test, it means the drug shows higher potency. For example, you have 2 drugs, one is with IC50 values 10 and the other with the IC50 values 50. So the IC50 value 10 is more potent or more better than the one which shows the IC50 value 50. Okay, then also we see PIC50 value. PIC50 value is actually the log of the IC50 value, the minus log 10. So when we calculate the PIC50 value from the IC50 value, then the values are became opposite. For example, I told you that the lower IC50 will be better. On the other hand, the higher PIC50 value will be better. If you don't believe, just take the minus log 10 of 10 and also take the minus log 10 of 50. As I explained to you in example, so the value for 10 will be higher than the value for 50. So it goes the same. Okay. Now, this was the introduction about the units, IC50 and the PIC50. Now we are going for some installation, and here we want to connect the ChEMBL web source client, and we also are interested to install RDKit. RDKit is a program used in chemoinformatics, and in such kind of studies we will heavily exploit the RDKit. Okay, now we will do some import jobs, which I did. Okay, if you run this or not, that doesn't matter. Actually I will run it. So in this step it will run, doesn't matter. In this step we will create some API tokens and to extract data, so you don't need just click them one by one. Okay, as I mentioned earlier, we will use the information of EGFR. So here is the trick. You will write your own ID here. For me, you see, EGFR human is 5005533. You will use your own. You can, but do not disturb these double dash, otherwise it will generate an error. You can do this way, like 500, but I will use 533. Run this. Now type this, click this, now click this. Okay, once we click this, it retrieves some data. This is important to understand. If you see, this data is almost similar to this one. The difference is, here we see index numbers like 0, 1, 2 up to 8, and here we do not have index number, just boxes, and so here we are. Okay, now we were interested in this single protein P00533, which is here, single protein. Also we have some another data for the single protein, so we can look both. Otherwise this one is also okay, but how we will know that which one is okay? So let's come down. Here is the index number. For example, you want to use this data, you will need to change this 0 to 1, but I will use the first data, index number 0. That's why I have put it 0. But in your case, because your protein will be different, so you need to type the index number which corresponds to your data of single protein. Okay, it gives some detail, like its organism is Homo sapien, protein name is this, target ChEMBL ID is 203. You can confirm from here, target ChEMBL ID is 203. Yes, we are on the right track. and target data type is single protein. Yes. Okay, we just to confirm, we just print like which data you are reading. It says ChEMBL 203. Again, you can see it is this data, and we are also interested in this data. Okay, now we are now importing some variables to the this notebook. Variables like activity ID, essay ChEMBL ID, and so on. So right now we are trying to import IC50 and the essay type. Essay type B means that they are binding in those essay. So the program will not will remove all those data which do not have IC50 or which do not have the essay type B. Okay, it will take some time, so let it be. Each entry in our bioactivity set holds the following information. So the program is retrieving the data here, and here we will print like what information this data contains. So let me make it pause because it may take a while. The data is too much. Okay, it took almost a minute. Now we click here, and it's gave us some information about activities and all those things. So if you want to read it, you can. Otherwise, now we are interested to prepare this data set and then download it. So this step should take almost 10 seconds or hardly 1 minute. Once you run this step, you will have I paused the session because at times it took more than 2 minutes. Okay, you will have the activity IDs and all other stuffs, activity essay type and ChEMBL IDs for different molecules. You can view this data. Already it should be present here. If you want, you can download right now, but let's wait. Now we want to know what are the unique units used in this data. So you can see there are too many different units, nanomolar, picomolar, molar, etc., etc. Now we will do some tweaking. The tweaking means because we want our unit to be kind of uniform. Now come here. This is the data processing and filtration step. We will remove the redundant data. Redundant data means the data which is present in duplicates or triplicates. Or we will also remove the data which has some missing value. For example, if they don't have IC50 value, it will not be worth for our project. So just run these commands. It will change some normal value to floating objects. our data frame contains this much of rows and this much of columns. These are the current units it contains. So we will run all these things and will remove some data. If you can see, the data became reduced to 6,000 rows because some missing data were removed. Missing data means that which is not completed in for all the variables we will need. Now we took our data, we took only those columns which we will need, but still you can see there is some redundancy. You can see ChEMBL ID 674637 is the same here, also this one. So this is called redundancy, but we reduce our column to IC50 standard units and units. Now 6822. Now we are mixing the bioactivity data with some molecular structures, because the 6822 means this much compounds in the data after filtration. So we want to include the information, the structural information in the form of smileys, which is the 1D representation of a structure to this data. So from here we have almost 6,822 compounds. We ask the program to combine the structural records from PubChem or from any other source to this data. So it will take some time because we have 6,000 molecules, so it may take 4 to 5 minutes. For the current time, I'm going to pause this session. Welcome back. So it is completed. You can see 100%, 6,822 molecules are downloaded. So if you analyze, or if you think, what have we done so far? We just fetch our data in the ChEMBL, then we remove some redundant data, some missing data, and finally we downloaded the compound structures present in that data. In my case, we had almost 7,000 molecules, so it took 30 minutes. In your case, it could be different, much higher or smaller. Now we will try to work for something. The data which we downloaded a little earlier contains 2 columns. Of course, one is for the compound name and the second one is for the canonical smileys. Here it is. These are the molecular structure in the form of smileys. We already talked about these smileys in our docking through Google Colab, and this is the column for IDs. Okay, here we have, if you can see, the 6,822 molecules were reduced to 6, 8, 15. Almost 7 compounds were dropped because of the issues with canonical smileys. Simply, you can understand this way. Or any duplicates were removed. Now what we are going to do, we are trying to fetch this data or to combine this data with the previous one. Okay, so this is our original data set. This is the filtered data set. We removed the redundant one. So just for validation, we ask the program to print it for us. Now let's merge both the data set. Output some data head. Now you can see, if you see here, initially our data was like this, but now our data is improved and we have incorporated structure for each molecule used in the bioassays. Now next we are going to convert the IC50 values to PIC50 values. I discussed earlier that what are PIC50 value. It is actually minus log 10 of the IC50 value. And we have also incorporated the data here. So you can see what I told you, that this PIC50 value is theoretically opposite. If you see, the lower IC50 is better, but here the higher PIC50 is better. Okay, now just some visualization. We just wanted to see the distribution of our data. So we had almost 6,815 compounds in this data set for EGFR, and based on the IC50 value distribution, almost majority of them are in this region. It means majority of them are better because the PIC50 value is higher. You can see here. Now we will use some RDKit objects and let's see what magic we are doing. We have sorted our data and asked the program to highlight for us the data which is highly potent. You can see I just mentioned 3 heads. If you change this 3 to 4, so you will see 4 results here, although the whole data is huge. Now Here we will save our dataset. The name is EGFR underscore compounds. If you want to change this name, you can change this word. It should come here. EGFR, here it is. So now finally we will download this data. And let me open it. This is PIC 50 column. Okay, these are the smileys, or I would say the structures. These are some units. These are the IC50 values. So and these are the ChEMBL IDs of the compound, and this is the index number. Okay, don't save it. Our data is present in the download section. Should be here. Yes, it is here. We will use this data in further sessions, and we will do much more things, some filtrations, some analysis, and finally we will also do some machine learning, and then we will also perform some predictions, and how in the meanwhile we will also learn how can we manage, or how can we screen our library before docking, and how can we make our library much authentic before docking. So that's it for today. In this first session we have learned how we can use ChEMBL, how we can import the ChEMBL data, how we can treat the ChEMBL data, and how we can filter the ChEMBL data, and finally we can save this data into our computer for further usage. If you want to be with me, and if you want to learn with me, so please subscribe this channel, and also turn on the notification button, so if I upload the next part 2, you will have a notification in time. Thank you so much. Bye bye.